Our first speaker today is Bharath. Bharath has received his bachelor degree from UC Berkeley in EECS and mathematics, and he was a valedictorian of his graduating class in mathematics. He did his PhD in computer science at Stanford, where he studied the application of deep learning to the problem of drug discovery. Bharath's graduate education was supported by the Earth Fellowship, the most selective graduate fellowship in the sciences. And he is currently the founder and CEO of Deep Forest Sciences, a startup building no-code platform to allow biologists and chemists to leverage scientific foundation models. He's also the author of TensorFlow for deep learning, from linear regression to reinforcement learning. Go check it out. And also a developer of the Introduction to Modern Machine Learning with O'Reilly Media. He's also the lead author of the Deep Learning for the Sciences, another book. Please go check it out, and please help me welcome Bharath to the stage.
Recording in progress. And if we share your space, and then the microphone first, second, good answer. I got it. Okay. Give me one second. Let me go ahead and
we should be able to do that now. I think so. Now you can try again. Yes, you're good to go.
All right. First, good morning. I'm really excited to be here, and this looks like an amazing conference. I think thank you to the organizers for inviting me here, and I'm looking forward to having a chance to get to know all of you, and also maybe to share a little bit about my experiences applying machine learning to drug discovery, and where it could be maybe useful to you all. So a little bit of background about myself. One second. There. I did my PhD at Stanford in the Pondé group, and when I got there, machine learning for drug discovery wasn't really a thing. So the reason I was excited was, I took Andrew Ng's seminar at the time, and he was saying, deep learning is really cool. It's going to change all the world, et cetera, et cetera. And I was like, oh, I'm going to go do cool things. But, well, if you've ever been to Stanford, there's a perennial problem there. The professors keep leaving to go do exciting startups. So every professor I thought about joining had basically left to do their own startup, I think including Andrew at the time. So I was kind of wandering around, and then I took to wandering in different departments. Luckily, I was supported by a fellowship called Hertz, which gave me a lot of freedom to do this. And I wandered into the biochem building, and then I saw these beautiful posters that had these pictures of like electrons. And at the time, I didn't know they were like electron densities. It was very colorful. It was like a rainbow poster. And I kept seeing this like 4 times. I was like, I need to go in and talk to these people. So that was Vijay's lab. So Vijay and his group primarily had gotten known for working on protein folding, like folding at home. These days, you might know him better as an investor, but at the time, they were getting into problems of applying computational methods to drug discovery and some early work on machine learning in drug discovery. So I talked to them. They're like, oh, we'll give you a free desk. And I hung around. And in time, I learned a little bit, And I think a lot of that, especially on the drug discovery side, really is thanks to, say, other folks in the lab who tutored me, and lots of conversations over, coffee, Wikipedia. There were no resources like, this entire summer school. So I think it's an amazing opportunity that, really, I think everyone in the ML for drug discovery team here has put together for everyone to kind of hit the ground running and learn about the field. So I'll try to distill, say, some of the things that, I've learned. I guess I've been working in the field for a while now. Well, but compared to some of the folks who really find drugs, I'm very much junior. You'll see that there, a lot of drug discovery is still gut intuition and practice. So I was looking forward to seeing what that impact versus hype slide would actually land at. My personal score, I'd say, was like hype 8, impact 2. And I think that's, despite all the energy, despite all the hype, machine learning is not that useful in drug discovery. I don't know what you want to hear at the start of a 2-week workshop. But part of the reason is I want to tell you that up front so you can change it. And I hope to explain why I think drug discovery in particular is such a hard problem for machine learning and why current methods are still very weak. They don't solve the actual problem. And maybe this will be a recurring theme. You come in, you read all these papers, you're like, oh, numbers going up, benchmarks are being broken left and right, AGI is coming. And then you try it on an actual problem and it's kind of useless. Like, this happens so much. And I think that learning to disentangle the hype and the marketing, especially, and I will step out of my hat as a startup founder, say, especially from companies who need to raise money, to actually what the scientific reality is, I think is a really powerful skill. So again, we'll maybe say a little bit more about that. And I think, hopefully we'll also, point you to a couple of resources that could be useful. So while I was at Stanford, I started an open source project called DeepChem. DeepChem is still going quite strong today. It's an open source community primarily of AI tools for drug discovery and other things. We construct these reproducible scientific workflows and pipelines. In particular, think of it sort of like a scikit-learn for applying machine learning to chemistry and drug discovery. Especially if you're new to the field, I think can be a really useful resource. I think a lot of folks get their first start with kind of hacking basic pipelines in DeepChem, after which, you start building your own things. So again, I definitely recommend checking it out. We're used very broadly by the industry. I think every organization listed here has used DeepChem for a paper or a blog post or something like that. There's a big active open source community. I'll have a Discord link later. So again, if you're just getting started to the field, I definitely recommend checking out DeepChem. We provide a lot of basic, simple tools up front. It's a very active open source community, can help you hit the ground running. And finally, I think one of the coolest things that we've maintained for a while now is we have a collection of data sets that's kind of baked into DeepChem out of box. So if we're getting started with machine learning and drug discovery, DeepChem and MoleculeNet might be your friends. Now, that said, I'll say a couple things about the caveats of using these tools later. But when you're getting off the ground, when you're just trying to get something working, check out DeepChem. We'll have more links later on this. Okay, with that little bit of background put away, I want to say a little bit more about the process of drug discovery. So maybe let me actually take a quick straw poll here. Like, how many folks here come, say, from a primarily computer science background, if I could get a show of hands? All right, I'd say maybe most folks. How many folks come from, say, a biological, chemical, medicinal background? All right, a lot of folks here. So I think a pretty even split here. So for some of you, this will be things you've heard before. Others, this might be a little bit new. But finding a new drug is really, really hard. And I think the biggest part of why it's really hard is that the body is extraordinarily complicated. We don't really understand what causes most diseases. And again, this is, I think, the first thing to kind of like really bake into your heads. Like, the cause of most diseases, with the exception of some, say, very well-studied diseases for which there's rare genetic factors or things like that, is very hard to pin down. What you often call a disease is actually probably something more like a cluster of closely related diseases that look like each other, but that have different causes. For example, you take something like Parkinson's. There's like some genetic causes of Parkinson's that cause is a strong word. Some mutations that predispose you to Parkinson's. Then there's this big word, idiopathic Parkinson's, which idiopathic kind of means like, I like to think of it like, I'm an idiot, I don't understand this, but it sort of means like no one understands it. Like it basically means here's something that looks like Parkinson's. And for a long time, the way you diagnose Parkinson's is you'd actually have to go to the clinic and take like a battery of like behavioral tests. More recently, I think that like the Michael J. Fox Foundation, I think they had a paper on a new biomarker for it. So there's a more, known way now of beginning to diagnose it. But this is the state of affairs for most diseases. In many cases, we don't really understand what's happening. We don't understand why it's happening. Doctors know a lot about actually what you see in patients. There's a lot of actual information known from observing patients, but the molecular mechanisms, the physical mechanisms, a lot of this is still up in the air. So when you're doing drug discovery, the first and hardest problem is actually picking what to actually try to do. So the way a lot of modern drug discovery works is you take this reductionist approach. You say, I can't boil the ocean. I'm going to pick this one protein or something. I'm going to say, can I turn it on or off? And if I can do that, maybe I can affect this disease. In practice, that's a terribly reductionist assumption. A lot of drugs actually affect all sorts of things. But it's a useful first approximation that can help you get off the ground. That's often called target discovery. I think some of the speakers, I think later in this seminar, will talk more about that. So I won't say too much about that. But once you pick that target, the next stage is you want to find, can I find some way of, messing with it? And what you do is often called, hit finding. Can I find a first molecule that has some interaction with this target in question and that, turns it on, turns it off, but at least there's a strong measurable interaction. And then from there you say, can I, make it more drug-like? If you have, say, there's a terrible example, like if you pour bleach on a cancer cell line, it's going to die. That's not a drug though. So you want to always make sure that the degree of, say, toxicity, the degree of solubility, all these factors are under control. And then, finally, after you do all this, then you need to do testing. You need to test it, say, in different models. Like originally, maybe you might do like a purely in vitro test, purely in a test tube, vitro as in glass. Then you might do in vivo, like that is in life forms, rats, mice, other animal models. Then you might move into the clinic. You actually test with human volunteers, and you have phase 1, phase 2, phase 3 trials. It's a very long process. Again, I think it's about maybe 15, 20 years, I think is not uncommon. Most even senior scientists who work in industry have not found a drug. There are a few people though who are actually very good at this. So this is part of like, there's this very mysterious art of like being good at finding a drug, and it takes 20 years and there's a lot of luck. So one of the weird shifts is that most people who work in drug discovery don't actually find a drug. Maybe you contribute a little bit to being part of it, but it's hard to actually measure contribution. And I think this is part of why, like, I think that if there's one thing I think AI is bad at right now, it's understanding long time scales. You look at ChatGPT, you go back and forth, you talk to it a little bit, but it's like a conversation. It's not this thing that spans years and decades. Like that's a very human thing to understand time at scale. And drug discovery in some ways is like similar to mega engineering projects or large construction projects, like those cathedrals or temples that take centuries to build or something like that. You hear oftentimes it takes 20 years for the drug to be discovered. You go back to the scientific history. You're looking 50 years, 100 years. So understanding the notion that time is involved, that there's a lot of like unknowns that are fundamental to the process. And at best, machine learning is like a very, crude tool that gives you a window and a look into one part of the process. I think that's useful to go into this side with like this problem of machine learning for drug discovery. Again, there's so much hype around, AGI is coming for us all by I think they said 2019, then they upgrade 2027, something, I don't know. I'll wait till Terminator shows up. But, I just think that there's a lot that goes wrong when you think about things at time. It takes effort and time to actually get things to market. Anyways, so with that hopefully useful, or maybe not useful, discussion of the field, let's talk a little bit about what machine learning for drug discovery actually tends to look like. So again, the first thing, as briefly mentioned earlier, I think this series, this summer school primarily covers small molecules. Small molecules are, look something like that. Or say, I don't know, 10 to maybe 100, 200 atoms large. They are, mostly molecules that are amenable to the techniques of organic and medicinal chemistry. So you'll see a lot of rings, you'll see a lot of linkers, you'll see carbons, nitrogens, things you've seen in a textbook. Maybe if you've taken an organic chemistry course, you've seen these things. When I'm telling people what I do, I'd say like small molecules are things that you can turn to a pill. That's a almost, I think, accurate way of thinking about it. It's not like a vaccine. It's not like an antibody. It's not a shot, but it's something you can take in a pill. Small molecules are very powerful and useful technology, but if you go to a lot of new biotech companies, they're actually really excited now about what they call new modalities. People have done a lot with small molecules. We've probably not scratched the surface of like the ocean of small molecules. There's a lot of them out there, but there's a lot of new modalities, things like antibodies, like exotic things with RNA that people are working on, like genetic gene therapy. Again, there's a lot of really cool stuff that's happening here. Also, if you are based in the US, like I think the Inflation Reduction Act has changed it. So I believe that it's more favorable for companies to go after new modalities as opposed to small molecules. But no one's quite cracked that problem of making anything else really into a pill. So I think that small molecules are here to stay. They're basically what medicine's been for a long time, but the future might look different. There might be a lot of different modalities. So something to keep your eye on, but I think learning small molecules will help you understand these other modalities too. So I don't think it's wasted time. So now, looking at a small molecule, how do you begin to turn this into something you can do machine learning on? One of the most, useful ways, and this has been around, I think, it's like to say since the 70s or 80s or something like that, is to take a small molecule and turn it into what's called a bit vector, a collection of zeros and ones. And the idea is that you take different subgroups of the small molecule, you run them through what's called a hash function, and the hash function points it at some index in this bit vector. And then you mark those indices where these functional subgroups are available with ones and everything else is zeros. So you get what's called a sparse bit vector. These are what are called circular fingerprints. If you go to DeepChem, you'll find them under circular fingerprints. They're really useful. This is like one of these like basic techniques that often just ends up being really useful in practice. So I definitely recommend, regardless of whatever fancy transformers you might use, check out, make sure you're doing baselines with simple models and simple fingerprints. Once you have a fingerprint like this, there's a lot of things you can do. Once you turn things into vectors, all the tools of machine learning come into play. You can do, I don't know, similarity search, you can do simple classifiers, you can do regressors, all these things that you might have heard about elsewhere in machine learning. But I think, about, I'd say almost 10 years ago now, folks started asking, well, these fingerprints are great, but we're seeing all these powerful things happening next door in the machine learning world with convolutional neural nets. Is there something we can do where you can apply a convolutional neural net to a molecule? And actually, one of the first things people did is they said, well, we have pictures in organic chemistry text, so can we just feed those into a regular connet for images? That tends to work pretty well, but more recently, I think folks have also come up with the idea of using what's called a graph convolution. Again, this is a very foundational idea. The notion is, and this took, I think, a number of papers to work out, and there's some very beautiful, I think, theory on this, that you can do a connection between what's called message passing and a graph convolutional network. So you can think about it as having, say, make the molecule with its atoms and bonds into what's called a graph, a collection of nodes and edges. Make each atom into a node, make each chemical bond into an edge. Then you can think about a graph convolution as having, say, at each atom, have some local vector that summarizes the atom, then have an update rule that combines information about your neighbors. And you repeat this update rule over and over again. And at the very end, you average out all the vectors for all the atoms. So there's a notion of gathering information like from across a molecule and turning into a single vector. So if you remember from the previous slide, we did this hash functions on functional subgroups. So there's this actual hard function that does the crunching of different subgroups into little vectors here. Instead, you can think of a graph convolution as like a soft version of this, where you have a soft way to summarize subgroups, but it doesn't need to be these hard zeros and ones. It can be floating point numbers. So it's sort of a fuzzy fingerprint. And I think actually one of the first papers on this topic called it a neural fingerprint. So again, these are really powerful methods, I think very much worth knowing. Again, there's lots of tutorials out there. Check out DeepChem. There's a lot of tutorials on how to use things like graph convolutions. Many of you are probably already familiar with it. I'd say in many ways, they're like the baseline for a lot of the machine learning methods that are happening in the field. And then, finally, I think that one of the big waves that has happened in the last, 3-4 years, something like that, has been applying transformers to chemistry. So the idea of a transformer is like, if you take a small molecule, there exists a standard language for these small molecules. Actually, there exists a few. It's often called SMILES, and more recently, people have been introducing these other languages called selfies, and I think there's a couple other variants as well. By turning a small molecule into a string that is a collection of characters, you can then pretend it's a sentence. And when you pretend it's a sentence, you can then apply transformers and other machine learning methods designed for natural language processing directly to the string representation. Now, you might reasonably ask, like, why does it make sense to treat a molecule as a sentence? And I can give you some made-up answer, but the real answer is like Hugging Face and some other folks made some really nice kernels for these things, and they run really fast on big GPUs. That's more or less it. Like, there's a tremendous amount of engineering work that's gone into building infrastructure for these string processing transformers. There's also a lot of theory, and I'll get into this a little bit later, about what's called self-supervision, pre-training. So this is a technique that, say, the GPTs or other transformer models use to learn, quote unquote, learn basic facts. So if you can leverage this intelligence, this textual intelligence that has been built elsewhere, and apply it to this field, maybe you can do something. And that's more or less the inception of why I think a lot of us started working on these questions with transformers. Again, I think a lot of, as with many application fields, a lot of what is done in AI for drug discovery is often driven by broader trends in machine learning at large, and you try to adapt these tools to the problems of drug discovery. Now, that said, there's often a lot of adaptation, and there's sometimes it's not a one-way you just take. There's a lot of ideas that I think have flowed back. But it's kind of beginning to say, can you leverage, say, what a broader community has built? I think is really a useful way to get some things built. Okay. So with that, introduction, let me then start talking a little bit more about what machine learning drug discovery actually looks like. So I think one of the fundamental things about machine learning drug discovery is it's a low data problem. And I think this is an intrinsic part of the field. Let me explain what I mean by that. Let's say you are a big tech company and you want to build some big machine learning model. Where do you get the data? Well, it turns out that there's basically businesses, I think primarily, I'd like to say India, Africa, a couple other places like this where there's these giant call centers. Folks sit down, you pay them to label images, label captions, look at Waymo's shots from their camera, label what's going on. There's all sorts of ways people do this, but you generate a large training data set. This is not really feasible in drug discovery. Unfortunately or unfortunately, you often need graduate or extensive technical training in order to even be able to run the experiments that generate this data. So it's much harder to generate data the way the rest of the field generates it. Now, there are, in recent years, I think, an increasing number of methods for, say, applying robotic. What I mean by that is like, starting from the 80s, people started working on, say, automation robots to run certain classes of biochemical experiments at scale. This started, I think, in the early days at like 10,000, 100,000 data points, got up to about like a couple million data points. In the last 10 years, I think people have figured out really clever ways to use sequencing technology to be able to, say, tag small molecules with oligonucleotides like DNA base pairs and stick that into a sequencer. So you can actually generate, in some cases, quite big data sets. But there's an illusion. Like some company can come in and tell you, oh, we have 1 billion data points from our DNA encoded library. Or, I love Recursion, it's a great company, but let's say you have a billion pictures of a cell line or multiple cell lines. And you might think, wow, that's so much data. Why have we not cured drug discovery? The problem is like the funnel. And all of this data is very, very early in the funnel. You're studying a particular cell line, you're studying a particular protein system. What you don't have is data from live human patients, and there's good reasons for that. You cannot test drugs on actual patients without going through an extensive safety process for the benefit of the patient and society. When you get down to actual data that's pulled from patients, it's much, much harder. In fact, if you had a lot of data on a particular drug in human patients, that wouldn't be a drug you can sell to people. That would be aspirin. It'd be something that's been on the market. And interestingly, aspirin has a lot of mysteries about it. So even if you have a lot of data, that doesn't mean you really understand the drug necessarily. So when you're designing a new drug, fundamentally, you are trying to create something new, or in some cases, maybe we're trying to make a generic version of something that exists, or a fast follower, as it's sometimes called. But even in those cases, you're changing something so it's different from what's already available on the market. And that by definition, that means that you don't have the data in humans upfront. You need to generalize. And this is the hardest part of drug discovery, like for machine learning, is that it's fundamentally a scientific problem. You're taking a leap of understanding beyond what is known in the test tube or what is known in basic science, and you need a hypothesis. You need something that takes one leap forward. And again, this is why machine learning, it's a fundamental out of distribution problem. A lot of machine learning is really great in distribution. I think, again, like ChatGPT has really popularized AI, but it's done so in a way that I think has given people these really bad ideas about how good machine learning AI actually is. What I mean by that is like, I'll just go out on a limb and say, I think these companies cheat on the benchmarks. You go out and you take a lot of the data that's on the internet, you stick it in, and when you've digested all of Wikipedia, and you're like, who was Oppenheimer? And then in the background, there's like, I don't know, the actual Oppenheimer Wikipedia article, and somewhere it's like, oh wow, that's so smart. I don't know who Oppenheimer was, for example. You can do the same for a lot of, say, topics in science. So something like ChatGPT sounds tremendously intelligent, but when you actually probe it on scientific information beyond what anyone knows or has published on, all of a sudden things fall off a cliff. And the reason is there is no scientific structure, there's no inductive bias that actually tells these models how to generalize to new theories. And again, this is where science is special. There's actual structured hypotheses that enable these techniques to leap beyond simple prediction. And this is what machine learning for drug discovery has to do. You need to have some reason why what you're predicting can actually generalize further out. So this is a problem that I guess everyone in this field has been working on for a long time. There's a lot of different approaches people have taken for how to build models that generalize. What I'm going to talk about for, say, like the next remainder of this talk is some of the work I've done or my collaborators have done. There's a number of other takes on this. I think looking at, say, the schedule, a number of speakers will be talking to you about things like equivariant methods. That's another really powerful, promising approach. I won't say too much about it, but that's, say, baking in symmetries from nature into models so you can actually make predictions. Really cool approach. What I'm going to tell you about is a few different methods that I guess over the last several years that I've been exploring for how you can try to crack this problem of generalization in drug discovery. And I think the basic idea comes about like how, given very limited amounts of data, given the need to generalize broadly, can you build models that actually take that leap? And again, maybe there's four broad classes of ideas that I've worked on personally. One, this is a newer idea, something I'm excited about. It's by using physical simulations to bootstrap models. This is very hard to do for some reasons, and I'll say more on that in a minute, but I think there's some new reasons why this could actually be a feasible approach longer term. There's ideas from what's called metric learning. Again, I'll say the idea roughly, it's sort of like do nearest neighbors comparisons. Like if you have a new drug, and for some reason you think it's very similar to an old drug, then you can maybe, triangulate and try to take a guess. It's not a bad strategy, actually. It can work really well sometimes. It has some terrible limitations too, but I'll say a bit about that. Another idea, you can do, say, what's called multitask learning. You can say, well, I don't know too much about, say, this particular drug, but I know, say, a lot about this other particular drug, or maybe I don't know too much about this protein, but these 5 other proteins I know a lot about. Can I combine all the information that's out there into one unified data set and use that for training? And I think, by the way, this is the core idea, I think, behind, I'd like to say GPT-2. One of the reasons why it became so powerful is it began to leverage multitask learning at scale. And then finally, I think the notion of like transfer learning or self-supervised learning. And this is, another approach that the GPTs and ChatGPT have made very popular. Can you use some notion of what's called pre-training, learning from a curriculum ahead of time, so you can actually make predictions meaningfully in the future? I think there's strengths and benefits to all these methods, but again, going back to the theme of this talk, none of them solves the problem. You shouldn't expect any of them to solve the problem. It's more like having, useful tools in your toolbox so you can break them out for different systems at different times. Okay, So first, I'll only spend a couple minutes on this, but this is a direction I'm broadly very interested about. And the notion is that computational science, physics, has spent a lot of time building very powerful theories in order to run calculations at scale. For example, with things like density functional theory, free energy calculations, molecular dynamics. These are really powerful ideas that have kind of come about in the field of computational physics. But there's often limits to predictive power in the real world. And there's a variety of reasons for this. Like for physical models, sometimes you need to make approximations. And by making these approximations, you limit the actual ability of the model to predict reality. But there's a new wave of science, I think, physics informed machine learning. I like to call it differentiable physics, whatever you call it. The idea is you say that, hey, these things that affect the accuracy of the physical simulation, they're like parameters. And if you squint at it, it's kind of like a AI model. So what if I take a gradient, stick it all on a GPU, and I learn these parameters from data? I think there's a lot of power to this new idea that you can keep most of the physical simulation, most of the structure about nature that it encodes, but only change a couple things here and there to fit it better to data. So then you maybe hit that sweet spot of having something that has the predictive power of physics, but maybe the actual, ability to fit known experimental data that machine learning has. And if you can combine the two, you might have something that's really powerful. So some of the work that I've done with kind of my team has been looking at differentiable density functional theory. So again, density functional theory, it's a way to, study the electron density of a system and use that to predict various quantum properties. There's a really beautiful theory about it. There's something called, these self-consistent iterations that you have to do in order to get the system to converge. And some really neat work that folks at DeepMind, folks at Oxford, and us more recently have been doing is saying, can you take this, can you put it onto a GPU, and then if you implement this all in some framework, can you learn the parameters of the, say, what's called an exchange correlation functional from data? Again, I think this is early work. It's a little too soon to really apply this to drug discovery at scale yet. But longer term, I think more physics, more ties to nature are one very powerful way to kind of cross that barrier and have something that generalizes in new systems. Again, I think really cool idea, little premature in practice, but something to look out for in the future. All right, so next, I think this is a different idea. This is the notion of what I say is like called metric learning. And let me say what I mean by that. Let's say that you have some experiment, and this experiment has given you 5 data points, 5 readouts. Say maybe you have a variety of compounds I've listed on screen here, and they're either blues or reds. And if you have a new compound, you might say, is this a blue? Is this a red? What is this new compound? Well, in the absence of any other information about the system, what's the best you can do here? And what you might say is, well, we saw earlier there are these things called fingerprints. What if I, turn each molecule into a fingerprint, a vector? Then I can do some type of vector similarity between the vectors and, my styrene oxide, maybe in the vector space, I don't know, it's closest to dopamine. This is a made up figure, so I wouldn't read anything to that. So maybe I'll think it's a blue. I'll make a prediction like that. It's actually not a crazy strategy, like if you do this. Basically, then the question is like, what notion of similarity do you define there? What prediction do you make? And why do you do this comparison? How do you do this comparison? Again, this can work, I think, really nicely in some cases. The challenge, actually, sorry, I only have one slide on this. The challenge, I think, with this type of approach is like the notion of similarity is context dependent. Two things that look very dissimilar can actually be very much the same depending on how it is you're measuring similarity. Practice, you have to know a correct notion of similarity. So I think a number of years ago, like the citation here, we wrote a paper on this where we got some version of this to work on some small drug discovery data sets. We're really excited at the time. We're like, oh, wow, this looks like an amazing breakthrough. We can actually do lots of these comparisons, be able to find, new drugs with very limited amounts of data. And we tried it out on a new system, and then things didn't work as well. Like the challenge is the notion of similarity, the notion of how you compare to different things. It is very context dependent. Now, there's a big field that's arisen, from our work and other folks' work. It's called, meta learning for drug discovery. Lots of people writing papers about it. You can check it out if you're interested. I think there's a lot of people, smart people, trying to make the idea work robustly in practice. But I think the challenge is that these notions of, like, again, what is the vector that you're turning a molecule into? And then how are you computing the similarity between vectors? These are kind of like two meta hyperparameters in some way. And if you pick them just right, you have a beautiful model. If you pick them wrong, it's not predictive at all. So, at some level, you've lifted the confusion up one level higher up. Have you made progress? Maybe. Again, it's sometimes useful. What I can say is that I've never used these methods in practice myself, despite, inventing one of them. It's something that other folks I know, though, do swear by it. So this might very much be like a taste thing, something worth knowing. So check out metric learning, check out the notion of, low data drug discovery. It can work. Sometimes it doesn't. It's a brittle technique. Again, I apologize to any meta learners in the field. I'm not attempting to insult too many fields today. It is a really fascinating technique. I actually really intellectually like it. I think it's really hard though. You need someone smarter than me to work it out. Maybe that's one of you. All right. So from there, I'll jump to the third idea in our list, which is, can you combine different data sets somehow? And one of the ideas that I think came about, some of the starting work I think was done maybe at MSR. I think researcher there, Rich Caruana, I believe, in the 80s, I'd like to say, on what was called multitask neural networks. And the notion there is you take a neural network and you say, make it predict 5, 10, 20 things at the same time. And then by doing this, I think, researchers at the time found that this could lead to improved accuracy in machine learning because you don't tend to overfit on just one thing you're predicting. There's a notion that you're learning something that actually meaningfully generalizes to multiple different systems. And if you can do this effectively, that could actually be a really, powerful way of combining data from different areas. So one of the first projects I did, I think, this was actually at Google a while back. This is before TensorFlow. So there's a system called Disbelief that they had. It was amazing for the time, but maybe really janky by today's standards, as it always is. So we wired up some terrible thing in there that did the simulation. And I think, for the time, it actually worked pretty well. This was a lot of compute. I think at one point we were like fifth on the team I was on was lucky enough to have a lot of compute credits for some reason within Google. So I think for a brief minute we were wasting more compute on this than YouTube was. It was a day and then they kicked us off. This was before YouTube used GPUs, so there's a reason for that. And we generated this plot, which we were very proud of. And this is a complicated, gnarly plot, so let's try to explain what it is we were doing. As you go right, you're increasing the number of different biochemical assays that you're training the model on. Each of the lines is a separate one of the assays that we're using as a readout. There's something like 200 assays. We picked 10 of them. And then what we said was like, how well does the model do on these particular 10 experiments when you train with these up to 250 other experiments about what happens? And what we found was that there was a trend line where some of the models did really much better. You can see there's like one blue model that jumps all the way up. There's others that actually do worse. But when you look at the average, you see this nice trend that goes upward. And we were really excited. Oh, okay, this looks like it's really nice. It's a generic machine learning technique. We just pull data and we begin to, tie all these things together. You can actually see an improvement in downstream performance. This is great. This is going to actually change a lot of things. Well, it kind of turned out this ran into a wall. And the wall actually was not scientific for once. It was the problem that we'd more or less scraped a large subset of the publicly available data out there. And we said, well, big pharma has more data internally. Maybe we could get some of that data. Turns out lawyers very understandably don't like sharing internal secret data. My internship ended, this project never actually got further than that. But luckily, there was a there actually was an effort, a multi-year project, it's called Melody, by a consortium of different pharma partners that did a tremendous amount of work on both the legal and scientific end. They pooled data across different pharmaceutical entities, and they got some neat results. I am going to pull one plot out of these results. Again, I recommend reading the actual Melody paper. I'm just trying to summarize. I will briefly kind of summarize like what we're seeing. And they do see some improvement. I'd say the improvement that they're seeing was, I think, better than what we're seeing because they did manage to run this at bigger scale, but it wasn't dramatically better. You say 5, 10, 5, I don't know, I think we're saying what, maybe 3, 4% on average across the board. I think they were saying, I don't know, 5, 10%, something like that. I recommend checking out the actual paper. I'm, pulling these numbers off from memory. You see improvement, you do see some gain. There's value to the multitask learning technique, but it's not this dramatic, 100% jump in performance. It's like 10%. So if you do a lot of work, you get modest benefit back. Again, that's often the story of science. It's believable. Again, I really am in awe of the Melody project. The fact they pulled off the degree of if you've ever done a legal negotiation with multiple parties, or I hope you haven't, it's terrible. It's not fun. I'm just amazed they pulled this off. So kudos to them. They did great science, but I think it also shows that it's at least what I would call modest improvement. So there's some power to the technique here. I think there's a really cool notion that they arrived at. And I think that I do hope the industry continues to work on data sharing, sharing more data sets, especially from the later stages of drug discovery, I think could have a big impact. It's something worth doing, but again, think 10% improvement. It's useful, it's worth doing. It's not going to cure cancer, unfortunately. Well, in the near term, 20 years from now, who knows? These things can compound. Now, that said, I will take a brief aside and just go back a little bit. This is not part of the talk, but I think this is something that it's a hopefully useful scientific aside. So the way we did multitask learning was that, the original version of this, we took these circular fingerprints that I mentioned earlier. We took all the molecules in these 250 assays, we encoded them in these fingerprints, we fed them in at the bottom, and then we had, 249 explicit outputs. And we thought, okay, this is really cool. We can do this. One challenge, though, is what happens when experiment 250 comes in or experiment 251? Well, I said, oh, well, you have to retrain the entire network. Hardware will get faster. It works kind of. It's really janky. I think that what folks at OpenAI, GPT-2, and I guess other groups as well in NLP came up with was that this notion of, I'd say, language as a universal, emulsifier. What I mean by that is that they said, well, instead of having 249 particular outputs, what if we put out a sentence and you can have commas and you can just add more things at the end of that sentence? This sounds like, a third grader idea, but it's really deep. It's really brilliant because it means that you can now combine an arbitrary number of data sets. So I think, again, there is a tremendous power to the idea that you don't need to stop at 249. You don't need to stop at 752. You can just keep on going and have as many outputs as you'd like. And I think that's one of the most powerful intellectual ideas that LLMs have really brought to the table. The notion that natural language can just merge all these different modes of thinking or data into one giant data set. Again, I think I love the idea. I'm an intellectual all of it. I don't think it's going to cure drug discovery, and I'll say more about why later. But again, it's worth kind of explaining that I think multitask learning has been tremendously influential, I think, for this current AI boom we're seeing. And it's part of, I think, the reasons why ChatGPT, et cetera, is so compelling, because they didn't, unlike this old janky architecture we built, they didn't have one output for every, article in Wikipedia. They just have text, and that's, I think, the power of it. All right. With that, maybe that actually brings us maybe to the last set of ideas, I think, from the that we've been exploring. And I think one of these ideas that we came about was that, after looking at all these different ways of, combining different data sets, generating synthetic data sets, there's a fundamental problem, which is that at the end of the day, getting experimental data is hard. Again, it's expensive, it's difficult to do in high quality. It's actually, by the way, really hard to merge different data sets from different time periods. If you have a different sensor, a different cell line, everything breaks. But there's actually this new trend that's arisen maybe in the last 15 years, where although it is very hard to get more experimental data, getting new structures of molecules that are likely to exist is really easy. And this is due to the advent of something called combinatorial chemistry. Again, beautiful work, I think, is part of, like, I'd like to say, last year's Chemistry Nobel Prize. The notion that you can have these modular composable families of reactions that more or less, can be stacked one on top of the other. This means you can create a truly enormous array of molecules. So if you go to a vendor like enamine, for example, I think you can at latest count purchase up to 50 billion molecules. Now, what I mean by that is, like, I think at max, like 10 million, 100 million of these molecules have actually ever been made. But for the rest of the molecules with, I don't know, 80, 90% probability, they think they can probably make it for you if you pay them. And that's actually an amazing achievement, because this means now we can operate in this vast virtual space of molecules that are likely to exist. And these structures, you can find them on the internet. They're actually just out there waiting for you to do things with. So there's a tremendous amount of unlabeled data. Can we leverage this unlabeled data somehow and use that for learning? So again, the other, I think, big idea that, again, with these various GPT methods that's really arisen is what's called self-supervision. The notion of self-supervision is that given a data set, you learn by effectively occluding part of the data. If you're, say, looking at a video, you might stop the video at a frame and try to predict the next frame. Or you might, say, cut out a portion of a sentence and ask it to fill in the blank. You might do some combination of it. Like, for example, maybe you just cut out a patch in the video over time and you need to fill in the patch. This is a slide borrowed from, I think, from Jan LeCun, one of his presentations. You can see the link down there. If you'd like to check out Jan LeCun's original presentation, which is very good. Again, this notion of self-supervision is really also, I'd say, at the heart of, like, the GPTs and the pre-training that they do on web scale data. And one question that several groups, I think IBM, ours, NVIDIA, several other teams have been working on is saying, can you apply these notions of self-supervision to chemistry? Leverage the fact that there are these enormous data banks of structures out there and use them to actually boost performance on actual drug discovery tasks. Again, one way I like to explain it maybe is, like, we're thinking about, as humans, if you're a chemist, you train by doing a lot of PSATs. You sit down, write a bunch of reactions down. You have to make, on paper reactions. You have to run them in the lab. There's a curriculum. So you can think about this, can we build a crude curriculum for chemistry that we can use to train these models? So there's a lot of, like, different notions of how you can do this. I'll talk about one that we've used a lot in our work. And the idea is that first, again, take the molecule, turn it into a sentence representation, smile string. Then, given this sentence representation, borrow techniques for self-supervision from natural language processing. And what this boils down to is effectively you're doing something like taking a molecule, plucking out certain atoms and bonds, and asking it to infill those atoms and bonds. Or alternatively, what you might want to do is, say, predict certain basic chemical properties. Like, if you have a molecule, you can actually find its molecular weight just by summing up, say, all the heavy atoms in the molecule. There's other basic properties that you can compute using, open source libraries like RDKit. Can you train a machine learning model to be able to handle this basic curriculum before you try using it for an actual downstream task? This ends up being a pretty cool idea. Like, I'll say it's not the only way people have thought about pre-training these molecules. A couple of other methods that I think are really cool. There's one number of ideas borrowed from information theory, in particular the notion of mutual information. So the idea is that, well, we're looking. This is a really cool paper. It says, if you look at a molecule, it has a 2D form, but it's also a three-dimensional entity. Known information about the three-dimensional representation and align it with information about the two-dimensional information by maximizing something called mutual information. Again, I won't say too much about this. I think it's a really cool idea. There's other variants that do things like, still treat the molecule as a graph, but align information between the local representation and the global representation of the molecule. There's a lot of cool stuff there, lots of really neat intellectual ideas. I think what I'll say is that I think a lot of the more graph and mutual information-based methods are a little bit further from practice than the transformer-based methods, probably because, all the transformer folks, we just tend to borrow Hugging Face or other libraries, so that makes their job easy. Whereas, mutual information folks are actually, sitting there writing custom torch kernels and things. So they're doing harder work. I think they'll get there. But for now, I'll go back to say a little bit about a paper from a few years ago. So this was a collaboration project. I think this was actually started by Seon, who's a DeepChem contributor in high school, interestingly, at the time. So he approached me saying, do you want, can I do this, thinking about applying this to Europe's, or I'd like to see maybe a high school science fair? And I said, yeah, sure, whatever, this will be a fun little side thing. But it actually got some really neat results. And one of the things that we did in this paper was that we looked at, again, a plot similar to the one I showed you earlier in the collaboration with Google, where we looked at performance on certain tasks we cared about, but instead of adding more assays, we just added more molecules. And one of the things that really blew my mind is we're seeing more or less the same behavior and the same level of scale. There are no lawyers in the way. Like, in theory, the molecules are out there on the internet. You can download them. You can just keep going. So this is something really, got me excited about applying pre-training to drug discovery. The notion that the fact, due to combinatorial chemistry, we have effectively infinite structures out there. This might actually provide us a way to tap into scale. Now, there's been a lot of work on what are called these, scaling laws. I use scare quotes there because they're not really laws, as I would call them, but they are empirical, say, rules about the ways models improve that people have tended to observe in practice. This is the first time I'd say that you could maybe say that you could have tapped into some of that scaling behavior you see elsewhere in kind of AI as a field more broadly and applied to chemistry. So again, it was really exciting to us. We kept working on it. That was a project called Converta. We did a successor a couple of years ago called Converta 2. Again, I think that one of the ideas for Converta 2, again, is we were doing everything on a shoestring budget. Converta 1, we trained on Colab. Converta 2, we got a little bit of donated GPU time. I think one of our collaborators, they had a very nice, CTO was like, yeah, sure, use a couple of our GPUs. We're not going to, stop you from it. So we managed to scrape some GPU time, but we had to be very smart about doing minimal tuning. So we found ways to tune the hyperparameters on smaller proxy models instead of on the bigger models we trained. Since then, since Converta 2, a number of new models have come out. I think in particular, NVIDIA, and I'd like to say, not Salesforce, IBM, I believe. Yes, thank you. I think someone was just about to say it. Have put out some really cool big models. I think definitely recommend checking them out. Some of these are actually open source. There's a lot of really great work. I'll mention briefly, we are working on Converta 3. It has been in the works a long time because, again, we're trying to do it very cheaply. But I think we have something really cool. I won't, spoil the paper, but hopefully you'll see it later this year. But again, what I'll mention is, like, when we did Converta 2, again, this is a couple of years ago, what we saw is that the transformers are about par with Chemprop, like the state of the art at the time. I think the most recent, big IBM/NVIDIA, and I think, I apologize if I'm missing any of the other big models out there, have actually done better now, as in I think they're slightly better than the GraphCons, but with a lot more work you have to put in. So it's still quite hard to use these techniques, but I think they're one of the few techniques out there where I actually think there is an effective ladder to keep scaling this. I think there will be diminishing returns at some point, but I think you can probably keep going, like, I don't know, 10x what anyone has done so far, at least. So again, I think it's going to be really cool. This is one of the areas I'm very excited about in applying, AI methods to drug discovery more broadly. But with that, maybe, I might be running a little low on time, so I'll just quickly skip over this. Some other projects we've done applying what's called active learning. So you have these like active discovery loops. This is a collaboration project from a couple of years ago on a small synthetic experiment. We found applying what's called an active discovery loop sped up discovery, things like 3 or 4x in the synthetic experiment. Other folks have done more automated experiments in that vein. I'll mention one briefly. One collaboration we have with some partners actually away from drug discovery is actually over in battery electrolyte design. Actually, for those of you studying small molecules, I definitely encourage you to look at small molecules in other industries. There's a lot of them out there. So, agriculture, batteries, semiconductors, all these fields use small molecules. There's a lot of cool things you can do with the methods you'll learn in the next few weeks. We did this really cool paper, helped the team at University of Michigan and some other collaborators do automated discovery of battery electrolytes. There's a few papers on this. I think there's a more recent paper that was accepted that has, I think, an upgraded version of this. Again, really cool stuff that I think that shows you can actually begin to see automated discovery loops. One of the reasons this project is cool is these folks built a robot to do automated testing of like, they had like what I think called cell pouches where they could actually check the electrolyte dynamically. We can't quite do that in drug discovery yet, but when we can, I think it's going to be really cool. And finally, I'm starting to run into the end of time, so I'll quickly jump ahead to a couple other things I'll mention. This is a line of work, not one that our group has done, but something that a lot of folks have been looking at recently, applying LLMs to chemistry. What I will say, summarizing this field, is that it kind of works. Again, like if you apply an LLM to chemistry, they do digest Wikipedia, basically. It will appear to understand basic chemistry out of the box. Now, experiments we've done internally, as of yet unpublished, show that though when you take these models and you apply them to new molecules you have reason to suspect are not in the training set, things fall off a cliff. It gets really bad really quickly. You can do some bit with in-context learning. It's not terrible. There is this new effect called in-context learning. There's some promise to it. I think there is some hope for using the big transformers, using these LLMs to do novel chemistry. What I'd say, though, is the hype in this subfield has maybe gotten out of scope with what it actually seems to be doing. You need to actually bake in more understanding of chemistry. And I think that's fundamental to actually getting LLMs or current technology to do well. So I'm very bullish on, say, the power of transformers. I'm not bullish on, say, taking GPT-4 or 5 out of box and expecting it to solve our problems. There's a lot of molecules out there. If you actually look at available molecular data, it's about web scale data. It's similar. And I think these companies are not mostly training on this data yet. That's kind of what you all maybe should figure out. Like, how do you bring in actual chemistry knowledge and structures into large transformers if you want to use them? But finally, I'll mention a couple of last things before I wrap up. Everything in the talk so far today has been predicated on the presence of data. Big data, small data, little data. But what I haven't talked about is what do you have no data? And as a company at Deep Force Sciences, we work on a lot of actual drug discovery projects. Our partners come to us and sometimes they say, hey, we have no data. What can you do for us? And for a long time, I said, well, I can't do anything. But in time, I was like, that's stupid. Like, humans do fine, like you use other clues that are available. You need a way to be able to bootstrap from additional data. And I think that's part of why I've become increasingly interested in the power of physical simulations. So again, if you don't have an answer for what do I do if there's no data available, you are very limited in what you can do in drug discovery. So you need to be able to break out of like the mindset of saying, I'm a machine learning person who only works in data. And I'm saying this for myself because it took me a very long time to be like, okay, there's no data, but still we have to solve this problem. And I think, again, keeping no data is a really important special example. I think is actually really important for machine learning. What do you do when there's no data available? And finally, just getting to some last conclusions. So I think I have a couple of conclusion slides. The first one, again, of different methods out there for working with low data. Right now, the ones I think I'm most interested in, first, transformers. I think there's a lot to be said for chemical self-supervision. I think that there's a effective way to scale these models for a couple generations, which I think will lead to really cool downstream improvements in performance. Again, there's a lot of power. That said, remember what happens when you have no data. That's hard. That's why I'm also very interested in physical simulation. Physical simulations can bootstrap in the absence of data. That's one of the really powerful things of physics. And that's why I think increasingly physics will be important. It's been important already, but even more important for drug discovery. But finally, I thought it might be useful for me to take maybe the last couple minutes and just say a couple of conclusions about things I've painfully learned at times working on actual projects. And the first I'd say is, don't trust machine learning too much. Like if your really fancy machine learning model tells you something and you look at it and it looks stupid, probably is, a lot of the methods out there, I'm very guilty of this, so I'm not casting shade on anyone. dramatically overestimate their performance on real-world data. Anytime you apply a machine learning model to a real-world system, expect to see a considerable to complete loss of performance due to out of distribution. And just understanding this and not trusting the machine learning. And again, for those of you from computer science backgrounds, bullet point 3, understand the biology and chemistry. When I got started, I thought of this as a way to get a couple NURBS papers. I was like, oh, I'm not going to learn chemistry. I'll just, do a couple papers and move on. It was a really bad mistake. I really wish I could go kick my younger self and say, take some chemistry and biology class. Otherwise, you'll be reading Wikipedia for 10 years and you still will feel stupid. I think it's really worth learning the basic science. Like if you want to understand, you need to understand the science. That's actually what will solve problems in drug discovery. Machine learning is a tool. Science is a way of thinking. You need science. Don't rely on machine learning. That's maybe the biggest thing I'd say. And then finally, again, just brief thing. We do this at our company. Like if you work on some of these things and think you might be interested in working with us, reach out to me. But here's some of the papers I mentioned. But finally, this is actually the slide I want to end on. So if you're interested in kind of getting involved with machine learning for drug discovery, definitely encourage you to join the DeepChem community. That's a QR code to our Discord. Definitely encourage all of you to, if you're interested, join in here. Part of what we do is we try to build high quality open source tools for drug discovery. Again, DeepChem is a non-profit, educationally oriented entity. The goal is, again, build open resources. So we have a lot of tutorials. There's, everything DeepChem is open source, has been for a long time. It's MIT licensed, so you can use it if you want to found a company, if you can use it if you want to do something else with it, whatever you like. Again, one of the things we've recently been putting together, we've been putting a open source textbook together. So I've highlighted the link on the website. So it's deepchem.io. You can find the downloadable version of the book. This is actually a collated version of our different Google Colab tutorials. We have about 30 different tutorials. A lot of the subfields that you might be working on drug discovery actually are covered in the tutorial series. So I think it's a really great free resource. If you join the Discord community, there's like 1,000 people on there. It's a great place to connect with other folks doing projects and to also just learn about, especially if you're coming from a non-institutional background, it can be really useful to have some community to belong to. We try to curate it so again, it's safe and we moderate it heavily. So, it's safe because there's a lot of students on there. So again, we'll just mention, check out DeepChem. Again, as an educational open source community, we try to make it high quality tools for doing drug discovery. And finally, thank you all for listening. Hopefully I wasn't too boring or didn't. If you feel the need to kick me for insulting a research field, please wait till I get off the stage. But thank you again for taking the time to listen. And I really do hope you stick with drug discovery as a field. I think it's a fascinating intellectual endeavor. But again, what I have to say, one takeaway, you need to understand the science. Machine learning is not a substitute for scientific understanding. And I'll just highlight that 3 times. I think you cannot machine learn your way to curing everything. It doesn't actually work. Well, that said, maybe AGI takes over. Who knows? All right. With that, thank you all.
Hello, testing. Great. We are a bit behind schedule, but I think we could take maybe 1 or 2 questions really quickly. So, okay, I'll come and pass the mic to you.
Thank you so much. Hey, everybody. I'm Vlad, CTO and co-founder at Bioptic. We are pure computer scientists / deep learning people, and just recently dig into the drug discovery. And my question is, you were talking a lot about approaches. I like them, most of all of them. So thank you for that. But the question is, how do we trust those benchmarks you were mentioning? I like the paper of Camberta. Basically, yep. I also, took a branch of it for myself. And I also read a paper of Melody where they combined like about 10 data sets from different big pharma companies. But when we scale the data, we flood more data points. And when we benchmark, we are just closer in terms of the data points. Like similar molecules may be found with a much more higher probability within the test benchmark. So, and how do you assess those methods?
Yeah, it's a really good question. I think a couple of quick answers. First is, as a in general, don't trust benchmarks too much. Like I see a lot of papers and they say we hit state of the art in this. They probably did, but I know how it's very easy to overfit. So again, any one benchmark should not be trusted. The second, you need to take a lot of effort trying to separate the training data from the test data. There's a lot of different scaffold splitting methods and other things out there. These sometimes work. I think it's a really hard question because I don't think there's like one answer. I think it's more like if you're a practitioner, be systematically aware every model is overconfident. Don't trust the model past a point. So when we apply things in practice, we always start with the random forest. We start with very simple, basic techniques, like even simple physics-based techniques. We always set a baseline and calibrate from the system upwards from there. And if we ever see where the machine learning, transformer seems to do a lot better than the baseline, we know we made a mistake and there's a bug. At best, you expect a few percent better. In practice, if you're writing a paper, though, there's a great blog post actually by, I think, Pat Walters critiquing MoleculeNet. And again, like I'm one of the lead authors on MoleculeNet, but I definitely recommend also read the blog post critiquing it. We do the best we can, but it's really hard to make benchmarks in this field for a variety of reasons. Again, this is a longer conversation that might merit actually like a sit down talk, but for anyone who's interested, again, check out scaffold splitting, check out ways of making sure test data is separated. You will leak regardless. Just, don't trust benchmarks too much.
Great. Thank you so much, Bharath. The slides are available now on the portal space. So you could follow up with that, learn more about DeepChem, join the community as well. And yeah, we have made most of the slides for day one are available now on the lectures and lab resources space. So if you want to quickly request access, we'll make sure you can get those before the lectures actually start. Thanks, everyone.
Thank you, everyone.